Welcome back once again, dear students. Today our topic is principle of dominance. And to understand the concept of dominance or principle of dominance, we will continue with the Mendel's monohybrid cross. We will use Mendel's monohybrid cross that he performed, okay, to understand the principle of dominance. First of all, what is a monohybrid cross? You already know. A cross made between two individuals that differ with respect to a single characteristic, and here that characteristic is seed shape. Okay, one individual possessed round seeds, another possessed wrinkled seeds, and then when Mendel allowed them to cross, he got the F1 progeny in which all the plants showed round seeds. Okay, these findings, these revelations were used by Mendel to arrive at an important principle of heredity, which is known as principle of dominance or concept of dominance. Why this is known as principle of dominance or simply the concept of dominance? Why not a law? Because the principle of dominance, it is not universal. There are some characters in which the two factors, they do not show dominant recessive relationship. For example, we have incomplete dominance and codominance that we will discuss in the coming classes. Okay, so the important findings that Mendel used, important revelations that Mendel used to arrive at the concept of dominance are, according to Mendel, an individual receives one of its pair of genetic factors from each parent. As a result, F1 hybrid received R factor from one parent and small r factor from the other parent. However, in the F1 generation, only the trait encoded by capital R allele or capital R factor was expressed in F1, while as the small r allele it was not able to express in F1. That is why all the seeds, all the plants in F1, they produced the round seeds. Okay, those traits that appeared unchanged in F1 heterozygous offspring, Mendel called them dominant, and those that disappeared in F1, Mendel called them recessive. So the concept of dominance says, in a heterozygous individual with two different alleles at a locus, one allele which is dominant, which is able to express, okay, it suppresses the phenotype of the other allele, which is called as recessive. Okay, that means the expression of the recessive allele remains masked. Now let's understand it more clearly. Two pea plants, one possessed capital R and another one small r. Okay, then at the time of formation of gametes, this will produce 4 gametes because meiosis leads to the production of 4 gametes, and since all these 4 gametes they possessed capital R, we can represent it just by a single gamete because they are all similar. And same is the case with this individual. 4 gametes will be produced and each of these gametes possess small r. So we can, for the sake of convenience, represent it by a single r. Okay, and then these 2 gametes after fertilization, they give rise to capital R, small r. That means the offspring. Okay, in the F1 generation, Mendel found that all the plants they produced round seeds. This is only possible when this R allele, capital R allele, it is able to express it is round seed shape phenotype, and this small r allele, it is not able to express it is wrinkled seed shape phenotype. That means in the F1, although both the factors are present, but only this capital R allele is able to express its phenotype. It is dominant. It masks the effect of this small r allele. It does not allow the small r allele to express its wrinkled phenotype. Okay, this interaction between capital R and small r, which does not allow the small r to produce this phenotype, because of this capital R, this is known as principle of dominance. That means in a heterozygous individual, the two factors of a character which are present on the same locus, one is able to express its phenotype and another one is not able to express its phenotype. Okay, this is all about the principle of dominance. I hope you all understand this. See you soon with the next lesson on genetics. Till then, stay blessed and thank you.